Hello and welcome to another tutorial by Get Ahead Institute. In this video, we're going to talk about cell cycle and cell division. So after talking about cells, we're going to have to talk about how these cells divide and what stages do these cells undergo in their life cycle. All right, so if you haven't subscribed to our YouTube channel, please do so and leave a like to help the YouTube algorithm. So let's get right into this. So basically, uh, the cell cycle, when you talk about the cell cycle, we're talking about the regular sequence of events that results into new cells. So, a cell cycle, we're talking about the cell starting from when it's just a new cell, all the cycles that are going to happen, its growth, and all up to its maturation and division, basically. So, that's what encompasses what is known as the cell cycle. So each cell will undergo this cycle, and basically, the knowledge of these different cycles helps in giving important information to people who are studying cancers, their causes, and how this cancer is progressing. Because cancer is an uncontrolled division of cells, basically. So if you can understand cell cycle processes and what is happening here and there, you can try to gain valuable information about different cancers. So the cell cycle in a nutshell has basically four distinct phases, which are uh, mitosis, G1, S phase, and G2 phase. Then there's also another phase which is not typical cell cycle phase, known as G0, and we'll explore why it's not a typical cell cycle phase. Okay, so the molecules that are responsible for controlling cellular division, or uh, not really cellular division, but cellular cycle, like which molecules are going to be responsible for determining where this cycle ends, where the next cycle begins, where this next stage starts, and where the next stage stops, is known as a cyclin. So cycle, cyclin should make sense. All right, so we can move on. So the cell cycle is briefly uh, elab- elaborated, or briefly uh, briefly briefly explained by this picture. Okay, so the cell cycle has the G1 phase I talk about, the S phase, we have the mitosis phase, and uh, G0 phase being there as well. Okay, so we have something to think about as well. Which of these phases is the longest? So you can just see by how many hours it takes for each phase to go through, for the cell to go through each of those phases. You can conclude that the G1 phase is the longest cell cycle phase. All right, so the cell cycle basically has these phases that talk about. G1, the cell is growing in size. The cell is undergoing a growth in size. It's taking in all of the material that it needs for its cellular functions and everything. Then when it reaches the S phase, this is where DNA replication has to occur in the synthesis uh, phase. S stands for synthesis. So there's DNA synthesis or replication. The DNA in the nucleus is being replicated in preparation for mitosis. Okay, so just after DNA replication, there's going to be a short phase known as the G2 phase. Then after the short G2 phase, we're going to enter mitosis. So in the G2 phase, the cell is just growing a little bit, okay, just to accommodate and to make sure that everything is in order before it can divide. In mitosis, the cell is dividing. After it has divided, those two daughter cells are going to go through this process again. Then there are those cells, the other cells that go into a phase known as G0, or quiescent cells, or differentiation uh, phase. For example, neurons, once they divide, they don't do these cycles again, they don't divide again. And so these neurons are going to remain there for the rest of your life. Since the time you're born, the neurons that are there are the ones that you're going to die with, basically. Those are cells that go into a quiescent stage. Okay, so there are some cells that can actually leave this stage when necessary. For example, liver cells or bone marrow cells. They are not constantly dividing, but when there is need to, like let's say you have shortage of blood, or let's say the liver has been part of the liver has been cut, it can actually grow back. The liver can grow back. Uh, the other half that is cut off. So those cells move from the G0 phase and then go back to the normal cell cycle until they reach here again, and then they're going to go into the quiescent mode again until that organ has fully been repaired. All right. So cell cycle checkpoints. In these cell cycles, you're going to find that there are checkpoints that you have to make sure that things are going properly. Okay, so the first one will be the G1 and the S phase checkpoint. Here we are checking if the cells have proper nutrition and is all of the DNA intact. Because after that, you're going to DNA replication. So you can't replicate DNA, excuse me, you can't replicate the DNA that is not intact or the DNA that is damaged. So if at this cell cycle checkpoint we find that the DNA is damaged, that cell will be uh, the cell will go into. I think I left some headsets. All right, so I was saying that we're going to check, and if we find that the DNA is not in good condition, that cell is going to undergo apoptosis or programmed cell death. But if it is intact, the cell is going to continue on the replication S phase. And then there's also another checkpoint that is uh, after the uh, G2. Okay, so there's no checkpoint at S to G2 because here is just a growth phase. But just before mitosis occurs, we need to check if the DNA has completely replicated after G2 and the M phase. Okay, so between these two, G2 and M phase, you're going to check if all of the DNA has been completely replicated, everything has occurred properly, and then you can move on into the mitosis or the cell division phase. Okay, this after this occurs, then uh, is has all of the DNA in the new cells been intact? Are all the chromosomes attached to the mitotic spindle? This is a checkpoint that happens during mitosis, like during the mid of mitosis. And when that is okay, then you can split the cells completely and you have two new cells that are called daughter cells at this point. Okay, so during the M phase or the mitosis phase, that's where cell division occurs, and that's what we're going to be exploring much of in this video. So during this phase, the cell will undergo what is known as cell division, and the cell division can be either mitotic or meiotic. Mitosis occurs in somatic cells, and meiosis will occur in gametes. So what's the difference between somatic cells? These are cells that are all of the cells in the body are somatic cells except the sex cells. The sex cells are known as gametes, and these ones are special because they have to undergo what is known as reductive division or meiosis. Meiosis is known as reductive division because you're going to be reducing the number of chromosomes in there so that you have at the end of fertilization you keep having the same number of chromosomes in the new organism. Okay, so before we go into cellular division, there's what is known as stem cells and tissue renewal. Okay, so stem cells are undifferentiated cells that reside under small populations in tissues and organs. The reason why this knowledge is important is because that's where we have the new cells that are going to be dividing and replacing cells that are dying. Okay, so they're going to be originating from stem cells that help in tissue renewal. So cell division that helps in tissue renewal needs stem cells. I hope I did not mix that statement up. But yes, there are tissues, certain tissues that need uh, new cells regularly, like for example the GIT. So those are going to need stem cells at the base of that epithelia that are going to be helping in this tissue renewal. Okay, so the divisions in stem cells are going to be called asymmetric. This means that one daughter cell remains a stem cell while the other cell goes on to differentiate. So basically what we are saying is that in asymmetric cell division, there is symmetric and asymmetric cell division. So uh, in asymmetric cell division, you're going to have a stem cell that divides into two, but then one of them is going uh, to commit to become a differentiated cell, while one of them remains as a stem cell. Because at the end of the day, the stem cell population needs to remain so that you can keep producing new cells at the end of the day. Because if all of them are differentiating, you're going to have terminally differentiated cells that cannot go back to produce new cells. So terminally differentiated cells usually do not undergo a division that is able to produce another cell. You need to go back to the stem cell for you to produce the same line of cells. So how do you keep that? It's asymmetric division. One goes back to being a stem cell, and then this cell can divide, and then one will continue, commit to become uh, will commit to become a differentiated cell by becoming what is known as a committed progenitor cell or transit amplifying cell. So that process of moving from a stem cell to committing to becoming differentiated, that interstage, the stage at which the cell commits, is called a progenitor cell or transit amplifying cell or a committed progenitor cell. That is what is happening in this slide. So cell division, we can now that we have talked about symmetric and asymmetric kind of division. So just notice for symmetric, you're going to have all of these cells, the two that divide here, all of them uh, are going to be coming down here and forming the rest of the cell line. So no cell remains to become a stem cell, and these are the stem cells that usually run out for organisms that, rather, sorry, for tissues that lose their ability to regenerate as time goes by. This is basically what is happening. Okay, so um, cell division now is the main focus, and we are going to go over these stages as quickly as we possibly can. So cell division, we have two types, mitosis and meiosis, and we're going to start with mitosis that occurs in somatic cells. And in this particular type of division, two identical daughter cells are produced that all have the the same amount of, or rather the same characteristics, the same amount of DNA, which is 46 chromosomes, and the stages as illustrated on that diagram, that we have prophase, and in some books will include another phase known as prometaphase. We're going to have metaphase, anaphase, and telophase as well. So what is happening in each of these processes is what we are going to go over just about now. So in the prophase, in this stage, the nucleolus disappears. Okay, the first stage, the nucleolus is going to disappear. That is the innermost part, that inner part of the nucleus. The nucleolus is going to disappear. So the chromosomes become coiled. That means they condense, and this is the time they almost start to become visible. Like we usually say that the chromosomes are only visible during cell division because that's when they coil up, and the coiling starts at prophase. Okay, so they condense, shorten, and thicken, and they now consist of two parallel subunits called chromatids, which remain joined to each other at a narrow common region called a centromere. But the chromatids cannot be recognized just yet. So this is starting to happen, and this is what we are basically seeing here. Okay, the process is starting, and in prometaphase, that's when the centrioles move to the opposite poles, the chromatids become recognizable, and then now you're able to see this even using a microscope. Okay, the centrioles move to opposite poles. This is the centrioles here, they duplicate, move to opposite poles so that they create the mitotic spindle in metaphase. So mitotic spindle, metaphase, mitotic spindle, metaphase. Uh, so some books will combine prometaphase and metaphase. Sorry, prometaphase and prophase. Some books will put these two together, but yeah, just know what is happening where. In metaphase, like we said, the mitotic spindle is formed. It attaches to the center, to the centromere, the centromere. Once it attaches, it pulls the mitotic spindles, which are the ones we talked about in the previous video about the cell cycle, sorry, the cell organelles, formed by the microtubules. As they form these mitotic spindles, these are going to be involved in movement of these uh, uh, chromatids to the opposite sides of the cell. So once they move there, in anaphase, that's where the movement is occurring to the different poles. And then after movement is completed in anaphase, in telophase, you have cells that are now, uh, sorry, let me just repeat that. So after anaphase, after the spindle fibers have moved the chromosomes or the chromatids to the opposite poles, the cell is now going to have to divide in a process known as cytokinesis. Cytokinesis is where the cell membrane of the cell pinches in the middle, forming a cleavage furrow that then uh, splits these two cells and forms individual daughter cells. Formation of two daughter cells with the same number of chromosomes as the mother cell. So the same number of chromosomes, same genetic material, same cell type, resulting from mitosis. Okay, that's basically what is happening here. So what I've explained in the other processes is basically what is being shown in this slide as well. So just to recap, prophase, prometaphase, metaphase, anaphase, and telophase. In prophase, you are having condensing of you have condensing of the nuclear material or the DNA, and then in prometaphase they become visible, and the centromeres, they're going to move to the opposite poles so that in metaphase they can form the mitotic spindle. After forming the mitotic spindle in metaphase and the mitotic spindle, after it attaches to the centromeres, in anaphase they're going to be moving to the opposite poles, as we can see there. And in telophase, at the end of it all, you're going to have uh, formation of the nuclear membrane as well. Okay, so formation of the nuclear membrane, then cytokinesis occurs where the two daughter cells are formed having the same genetic material as the mother cells. And that is what is happening, what is being explained there as well. So that 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 is basically it about mitosis. And lastly, we're going to talk about meiosis. Meiosis is also known as reductive division. This is of particular interest, especially going into the next topic that talks about gametogenesis, processes that occur in spermatogenesis and oogenesis. So in meiosis, this is known as reductive division because we are reducing the number of chromosomes at the end of the day. And instead of two, like in mitosis, the daughter cells will be four, and they're going to be genetically different from each other, all of them. They're going to be genetically different even from the parent cell as well. So the meiosis type of cell division is divided into two stages. These stages are denoted meiosis 1 and meiosis 2. So meiosis 1, meiosis 2, what is happening here is firstly in meiosis 1, we have prophase as well. And this prophase is also divided into five stages, which is leptotene, zygote, and packeting and diplotting stage of meiosis 1. So the process of the first meiotic division is very long and complicated, and it is divided into the following 5 stages. So in the leptotin stage, the chromosomes, as in mitosis, appear as slender threads here. Although each chromosome consists of two chromatids that are joined at a centromere, the chromatids are not visible at this stage. So at this point, just like in mitosis, the chromosomes are not yet visible. And then the second stage of prophase 1 is what is known as the zygote stage. In this stage, the lengthwise pairing of homologous chromosomes begin. Okay, so there is pairing of homologous chromosomes in the zygote stage. Okay, so this event is called a synapsis. So synapsis occurs in the zygote stage, and a synapsing pair is known as a bivalent. So a bivalent is where you have two homologous chromosomes pairing up, and then they form that pair, homologous pair. We're calling that synapsis, and it happens in zygote stage. So please don't forget that. They might give you an MCQ that synapsis occurs in which stage of prophase. We're supposed to say zygote. And then in the packeting stage, which is the third stage, it's very long and may extend for even for years. It is characterized by the following changes. The chromatids of each chromosome become visible separately. Okay, the chromatids for each chromosome become visible separately. So, like we are seeing there, you can see chromatids of each chromosome. So, this is an entire chromosome, while the chromatids are these single sides there. That's what they're calling chromatids. So, they become visible. Here, you're just seeing it as one single thing, and it may appear as one single thing under the microscope, but here you're going to find that they're going to have that characteristic shape of a chromosome that you're able to distinguish that has two different chromatids. Okay, so there are two central chromatids and two peripheral chromatids, one from each uh, chromosome. Okay, central chromatids and peripheral chromatids. So, excuse me. So, at this point in the packeting stage, there is another this can be a take home message for packeting. There is what is known as crossing over that happens in the packeting stage. So crossing over is where there is exchange of DNA material between two uh, two chromatids, so that we have genetic variability. That's the reason why the two siblings are not entirely identical, even twins themselves, just because of that genetic variability. Okay, except if the twins were there are those what are known as identical twins made of the same, dividing from the same zygote. But those that are fraternal twins, as long as you're siblings, fraternal, you're going to have different genetic makeup simply because of this process known as crossing over. There is exchange of genetic material that is happening. So if you have a chromosome like that and another one like that, they're going to exchange pieces of material, and at the end of the day, you're going to have chromosomes that are combinations of different traits altogether. Okay, remember that one chromosome is from the paternal side and one is from the maternal side. So where the crossing over now occurs, you're going to find a structure like this, for example, two chrom- uh, two chromosomes, one from the mother, one from the father, and they exchange material here. And this point where they are uh, cross-linking, okay, is known as a chiasma, or chiasma, however you might choose to call it. So that chiasma is the point where there is meeting, and then the DNA material is exchanged there. So that means that's how you end up having children that have traits from the mother and from the father combined because of this process that occurs uh, here. So basically, you're going to have uh, yes, crossing over and exchange of uh, material there, so that the kids have an assorted kind of genetic makeup. So at the end of the day, you find uh, siblings that have some are short, some are tall, all of that. So that's the genetic variability is coming about due to crossing over that is happening in the packaging stage. So just to recap, uh, leptogen stage, you're having condensing. Zygote, you're having synapsis. In packaging, you're having crossing over, and then in the diplotin stage, you are having uh, uh, what's this? You are having the paired chromosome forming what is known as a tetrahed, and this tetrahed is going to separate. Okay, so this is the process that we are talking about. Crossing over synapsis is happening like that, and then you have exchange of DNA material like that. And then the chromosomes, after they have exchanged this material, there's a process known as diakinesis. Okay, diakinesis is where you have these uh, paired homologous chromosomes. Now they're going to detach from one another. So at the beginning, we have pairing of homologous chromosomes at prophase. Okay, and in prophase, the first stage, which is leptotin, that's where we're having you're having a pairing up of homologous chromosomes. So in the last stage, you have diakinesis. In the diplotin stage, you have diakinesis, and diakinesis is where the strands separate instead of what is happening in the first step. So we are reversing whatever was happening in the first step at this point. And then after this, you have metaphase. So what we just from concluding there is prophase 1 of meiosis 1. And now going into meiosis or meiosis 1 metaphase, the second phase. Okay, so please don't get lost. We were in meiosis 1 and in prophase, and we explored the different stages of prophase, and now we are moving on to metaphase. Okay, metaphase is the homologous pairs of the chromosomes become arranged at the equatorial plane of the spindle. And at this stage, we quite know that once the mitotic spindle is attached there in metaphase, and in anaphase, the homologous chromosomes are going to migrate to the opposite poles of the spindle. Unlike mitosis, the chromosomes move randomly. Okay, the shorter chromosomes move earlier than the longer chromosomes. This is just the fact that you will put at the back of your head for the cases of MCQs. Then in telophase, at the end of the day, we have nuclear membrane is formed around the polarized group of chromosomes. The cell membrane constricts, and two daughter cells are formed through cytokinesis, which is the division of the cell membrane. Okay, so each daughter cell thus formed contains only half the number of chromosomes or haploid number with exchanged genetic material. Okay, so in meiosis, at the end of meiosis 1, this meiosis 1 is known as reductive division, because at the end of it, what you're going to be having is half uh, half the chromosome number, and that happens due to the separation that we had. Although these strands are still going to be double, they're just duplicated. That is what is happening here. Okay, so these processes move on like this. So in meiosis 2, the second meiotic division is essentially similar to mitosis. Uh, why is it similar? This one is uh, this one is done so, or rather, the second meiotic division is said to be similar to the first, or rather, it is said to be similar to mitotic division because you're going to have essentially the same processes occurring, but the only difference is that in mitosis, DNA duplicates, but in meiosis, DNA does not duplicate. So the reason why the DNA does not duplicate is so that when you separate those strands, you're separating each strand is going to be ending up in one cell. Okay, so when you have strands that are like this, at the end of meiosis 2, you want these strands to be separated, okay, each of them going into separate cells. Whereby, if it's mitosis, these strands are going to duplicate, and once they duplicate, you're going to have the double stranded chromosomes, and then these chromosomes are going to separate. That means you're just separating copies of each other. But here, since the copies are not made, you're separating the strands themselves. So at the end of the day, you're having two cells that just have strands, okay, that just have separate strands at the end of the day that each contain half the number of chromosomes, basically. And then just to briefly recap, these will be the differences between meiosis and mitosis. So mitosis takes place in somatic cells while meiosis takes place in germ cells. It's completed in one sequence while it's completed in two sequences for meiosis. For mitosis, crossing over of chromatids does not take place because you don't need genetic variability in somatic cells, but crossing over occurs in chromatids in meiosis, particularly in the prophase, and particularly crossing over will occur in the Parkinson stage. And daughter cells have the same number of chromosomes as parent cells, but here in meiosis, the daughter cells have half the number of chromosomes as parent cells. The daughter cells are identical in mitosis, while the daughter cells in meiosis are not identical to each other or to the parent cell. There is what is known as equatorial or equational division here. That means although cell division occurs, the number of DNA, the amount of DNA still remains the same. But in meiosis, reduction division occurs whereby from diploid you have haploid daughter cells being produced. All right, and then that is the end of this video on cell division and mitosis and meiosis particularly.